Hey guys. It's so dark, my camera. Everybody have a good weekend? I got a question I'm posing to everybody, so make sure you hop on there and fill it out. The link is in the chat.
Wait, Dr. Garneau, what do you mean by SOAP? Is that like an acronym?
No. So you must like cilantro.
I do.
Okay. Yeah. So when I have cilantro, it tastes like soap. So it's like this fight, this battle against Chipotle that I have because I like Chipotle, but I can't have the rice.
Right. Okay.
But good question though, because I use a lot of acronyms. For everyone just joining and flicking on the audio, as you're putting your name in the chat attendance, can you guys click on the cilantro inquiry for me and make sure you score yourself? There's 2 columns. Well, there's 3, one for your name. 0 if you, so if you, when you eat cilantro, if it tastes like soap, like it does for me, then you can code it like I did. So delish versus like soap, right? So 0 and 1 is how I coded it. If you like cilantro, you would code it 1 and 0. So if you guys can click on that link at the top of the chat and score your feelings about cilantro, then we can do something together.
Yeah, if you join after you send the link, then the link's not in the chat.
Oh,
so.
Thank you. All right, I'll send it again. I did not know that. Morning, morning. Wow. There's a lot of us that don't like cilantro in this class. Yeah. Somebody have a question? I'm gonna paste it again.
Sorry, I was just listening to music, but why are we deciding if we like cilantro?
Oh, we're gonna do an interactive math calculation.
Oh, okay. I was
Super
just like, I
fun.
don't know if this is like just a question we have.
No,
at the
no, no. No,
class,
there's a reason. So
Okay.
for people again just joining, I put a link in there to a Google spreadsheet. If you could code yourself based on whether or not you like cilantro or it tastes like soap. Okay, so if you click on that link, you should see the columns. And if you like it, you would code 1 and 0. If you think it tastes like soap, you code 0 to 1. I am a it tastes like soap person. So you can look at mine. If you think it tastes like soap, then you would do a 0, 1. If you think it's delish, then you would do a 1, 0. Okay. So click on that link. I'll post it again for those just joining. And make sure you code yourself in. All right. We'll keep that going. Give everybody a chance to sign in. We're evening out now, it seems. Okay. Again, for anyone just joining, make sure you go to this link and code yourself. Okay, we'll do a math problem together. All right, so I don't want to take up too much time, but I do want to make sure that I go over some logistics with you guys. Okay, so I'm going to share screen. And Gianni, if you can hop onto the link that I'm posting in the chat and code yourself about cilantro. Okay, you guys see the cilantro screen so far? Okay. All right. So I'm going to go to our Moodle, which I guess I have lost. There's so many tabs up here. Okay. Okay, so the first thing I want to remind everybody is that after class today, you can scroll down to the bottom of our site and access quiz 2. Okay, so click on the quiz 2 down there. You'll be able to access it. You'll have 30 minutes once you open it. It's 10 questions. You have a day to do it. So, you know, if you have lab or you have to work or you just can't fit it in, you've got until 11 o'clock tomorrow to do it. Okay. As I mentioned, content's going to include the animal adaptations, the life history stuff, and the demography, the life table population related material. Okay. So don't forget that. All right. Make sure you guys get to that quiz at some point in the next 24 hours. Okay. All right, second thing is we're going to work through our genetics. I know we got partway through and then I gave us a quick example on how to do the calculations, but I want to revisit that today. Make sure you guys are comfortable with the homework. So in the unit on population genetics, you'll notice there is a link below it for a Hardy-Weinberg homework, okay? This, if you click on it, is what it's going to look like. So there's going to be some questions. Most of them will be organized this way, where I'm going to give you like the total number of people we sampled or total organisms sampled, and then either give you kind of the, you know, this many were dominant or this many were recessive. This many had this trait, which is dominant or was recessive. So this first one is, you know, field biologists and their reaction to poison ivy. So if they get this kind of oozy reaction, which we know is a recessive trait to poison ivy, then determine how many are homozygous dominant. Remember, homo is same, dominant would be the big A, right? So we're looking for, the only question here is the numbers big A, okay? And so given these two values here, the total 155 and the 90, the number recessive, you need to work through that calculation. Again, we're going to review that today in class. The second question is also a recessive. You would approach it the same way, okay? You would have, you know, your total 145, and you're going to have 35 of them that were recessive, and you'll work it the same way. If you kind of go through, you'll notice the next two are dominant, so that has to be approached a little differently. And then the fifth one again goes back to recessive. So the first, the second, and the last one can all be approached the same way, just like we're going to do together with the Amish example, the Amish cardiovascular issue. The dominant ones, there's just one extra step, and then you proceed just the same way you do with a recessive. Okay, so we'll do that example, and that's with John Travolta. So you'll just, you know, have to kind of refer back to that as you go. This homework assignment, you can turn it in anytime this week would be ideal, right? And that way I can kind of catch it if you guys are struggling with any of the content. All right. Again, I'm going to just paste in for those who are entered a little late. If you can just fill out your preference on cilantro, the link that's in the chat, that would be great. Okay. All right. Any questions?
I did one of those.
What was that?
I was gonna say I did one of those like 23andMe DNA tests, and
Oh, yeah.
it like had something on cilantro, like suggesting if you like it or not.
Oh, interesting. I haven't
It
done
said
that yet.
Yeah, I did it. I did the like all the health things, and like it said some interesting things, like it's like, oh, you're like increased to get like Alzheimer's and a bunch of weird
Oh my
shit.
goodness.
But,
Yeah,
like,
I
it's
think that's part of why I'm hesitant. I don't know that I want
you
to.
can like deny to not see that, but like it had a bunch of interesting things, like, oh, is your second toe longer, and the
Oh,
cilantro
wow.
thing, and just like, do you have a widow's peak, and a bunch of
Yeah,
stuff.
yeah. All these Mendelian traits that we've been talking about.
So yeah, so it's really interesting to see all that.
Very, very cool. And that's Kaylin who's speaking.
Yeah.
Yeah, very cool. I might have to look into that. Neat. So you guys are familiar, right, where that assignment is. You can get your practice. We're going to go through some of that again today. And so let's not wait on that. Let's just hop right into that right now. Okay. So remember, we started that lesson on genetics off by talking about, about our two alleles, especially when we're looking at humans, right? Our two alleles at each locus, one from mom, one from dad, we get, and again, coding for our genotype. We talked about the two alleles, one, the dominant, the recessive allele coming together. Two ways to be dominant, right? Homozygous dominant, big A, also heterozygous, a carrier, big A, little a, right? Anytime you have that one dominant allele, you are dominant phenotypically, okay? Recessive, only one way to be recessive, little a, right? We gave some examples here. We did a Punnett square, right? Anytime you have, that one dominant, you're gonna look dominant, whatever the dominant phenotype is. Anytime you have two recessives, you're going to look recessive in terms of the phenotype. When we look at the combo potential at birth, right, 50% of the time the offspring, could be represented having that dominant, 50% having the recessive. We also talked about the phenotype, kind of the contribution to the environmental effects, right? That you could, think about genetic, the twins, right? Twin studies, twins raised together, same environment versus twins raised apart, that they have features that are, a little bit different. So phenotype can be influenced by their environmental conditions. Think about nutrition and so on, right? So we gave the example here with Daphnia raised with predators versus not, and how their size shifted a little bit, a way to perhaps avoid predation. We mentioned clines. We see this kind of traits along a cline latitudinally. There's variation in the environment along their range, right? We would expect them not to look exactly the same. We also talked about genetics, you know, and all the ways that we can have variation introduced, ranging from natural selection to random mutation to, you know, crossover when the alleles align at the metaphase plate. And we talked about mutation, which often happens randomly. And specifically, we gave some examples of, you know, a neat example of speciation that involved organisms in the same place, right? We talked about those salamanders, right? And this idea of macro mutation where, you know, normally when you look at the gametes, there's separation, you know, the alleles will separate. And one, you know, mom and dad will give one of each of those alleles to their offspring. What if that doesn't happen? What if mom gives 2 and dad gives 1, right? We have an increase in ploidy number, and that can arise in a new species. And that was the example that we gave here with the Tremblay's salamander, right? It was tetraploid instead of diploid, okay? We also talked about genetic drift. When population sizes get really small, and inbreeding tends to occur, we tend to see a shift in the number of deleterious or bad mutations that can build up. We mentioned that these small population sizes can arise from, you know, small groups of individuals moving on to new areas, and then they don't have any around, so they tend to inbreed amongst each other. That's founder effect. Bottleneck effect would be an example where some natural disaster came through and wiped their numbers down to something very small, resulting in increased inbreeding. We talked about that being counteracted if we can connect them somehow and have gene flow or migration, right? I gave some just cool examples of, you know, genetic, you know, anomalies and things that have kind of shifted traits through a population. The one example with the Seneca Falls military base, right? So we're in kind of central New York near the Finger Lakes, and we've got a military base that has this interesting deer coloration. It's a genetic mutation. They've been protected. Hunting any of the whites has kind of been prohibited. And as a result, we see the frequency of that allele increase over time in this population, which is not normally what you would see, right? You would think like a predator would totally take down more of those white deer, right? You think about the background and the coloration like we talked about, right? Let's see. We talked about the same sort of example with the mice and their background coloration, right? A mutation can arise. It can result in them having, you know, negative survivorship. And you wouldn't expect that to kind of be a trait that you would see continuing in the gene pool over time. Should be removed, right? If predators keep attacking and removing them from the population, there's fewer of them to mate and continue passing that trait on over time. However, there, you know, it really does depend on other environmental factors, right? Maybe it makes them have some kind of additional benefit, maybe not. In the case of what we saw here with these mice, with these like, you know, pretend Play-Doh type mice, any that didn't match their background immediately got attacked by some sort of a predator. You could see the marks on them, right? And again, in most cases, inbreeding is bad, right? We talked about the deleterious buildup of mutations over time. That's inbreeding depression. We see that with kind of the Florida panther, but outbreeding depression, where we would have migrants coming in to kind of mix up the gene pool, could also be a negative thing. And we mentioned that population could lose the locally adapted traits so that their offspring are timed with the right quality food that's greened up and so on, right? Just other cases of cool genetics things, the isolated wolves and moose out on Isle Royale, you know, disease coming in and knocking that population of wolves down to a very low number. They're not effective predators anymore. It results in the moose population exploding. And when the moose population explode and we've only got like 2 wolves, we don't expect the wolves to be able to make it, right? There's too much inbreeding going on. Unless an ice bridge forms in the winter, and we know that things aren't really getting cold these days, right? Except for now. anytime this can warm up, that would be great. But if it were to freeze over, which it does, every say 5 years or so, there's a potential for wolves to kind of walk their way across the ice bridge and infuse, a little genetic variation if they were to find and mate. However, the case is not looking good. So we expect that moose are going to continue to grow and grow and grow until their numbers overreach their maximum in there, and they've basically eaten all the good food and they start to starve. So we'll see their numbers decline as the food quality declines. So we left off here with Hardy Weinberg, and this is where the examples kind of fall into place. We're going to walk through the cilantro together. So we've got Hardy and Weinberg, these guys, mathematician, geneticist, kind of working together to model what we would predict, what we would expect genetically. So remember the 3-to-1 that we talked about with our squirrels, right? So along those lines. So these are the things you need to commit to memory, right? We've got 2 major equations. We've got 2 alleles. So we would expect in the allele frequency equation, there should be 2 variables. There's the P, the dominant allele, and the Q, the recessive allele. So P plus Q equals 1 is the allele frequency equation. Now, when we think about genotypes, we think about how we can arrange those alleles. What do you get from mom and dad, right? You could get big A, right? And if big A codes for P in the allele frequency, then P is big A, right? Big A codes for P times P, or P squared, okay? And then we've got a heterozygous, so you get 1 allele from mom, 1 from dad, a dominant and a recessive. That's the P and the Q. And there's 2 ways you can get it: P from mom, Q from dad, or vice versa. So that's where the 2 comes in. 2PQ would be your genotype frequency for heterozygous carriers. And then your Q squared, right, is little a times little a, right? Q squared is your frequency of your recessives, homozygous recessives, and that's where the Q times the Q, or the Q squared, comes in. So P squared plus 2PQ plus Q squared equals 1 is the genotype frequency equation. There's 3 genotypes that derive from those 2 alleles that we got, 3 ways that they could arrange, right? And those equal 1, okay? So we're going to use those equations to help solve those problems that you have on the homework. Now, population genetics, right? There's some assumptions for Hardy-Weinberg's principle. There's this, assumption that we will be in equilibrium genetically. Your allele frequency P and Q will not change over time if these conditions are met. So think about this like, in real life. Think about if you think this would really happen. So if we have a large population, okay, that's one assumption. Random interbreeding. So you can't have a type. For example, don't tell my spouse, Jason Statham is my type. I don't know if anybody else out there feels that way, but that's my type, right? So imagine kind of the mating scenario that you're randomly versus having kind of a preference, right, for mating. No mutation. We know that happens randomly all the time, right? No gene flow. Are we all fenced in, right? No, we can move around, right? Except now, right? We're kind of trapped by the border restrictions. So in this case, that's kind of an interesting thing, right? And then no selection, no natural selection. So those are all the assumptions of Hardy-Weinberg's model if our genes are at equilibrium, okay? If there's any sort of violation of that, of any of those, we can have evolution occurring, right? By definition, evolution is the change in allele frequencies over time. If we look generation upon generation and our P and our Q are always 0.5, then yes, we could be at Hardy-Weinberg equilibrium. But over time, if they shift, if those, if it goes 0.7 and 0.3, or 0.8 and 0.2, then we're seeing evolution occurring, right? The allele frequencies are changing over time. So those are the violations, right? It would be all the opposite, okay? And I did mention genetic drift already, but again, just some visuals here. The Amish would be an example of a group of individuals moving to one area, kind of tend to culturally and religiously kind of tend to mate together. So you would imagine there might be an increase in certain genetic predisposition for some sort of disease or syndrome that pops up in the genetics of the population of Amish, and that's true. I don't know if you guys can see down here this picture, polydactyly. You've probably heard this story, right? But some Amish families, there'll be a higher instance of children that will be born with extra digits, right? Extra fingers and stuff. And that's a part of what we're talking about. And then bottleneck, again, reducing both of these, reduce their population size down to something small, where there's a buildup of, inbreeding effects as a result of that. Those are genetic drift examples, bottleneck and founder effect. Okay, so again, over time, natural selection, you guys are familiar, right? Over time, there's some sort of genetic trait that allows you to survive better, gives you better opportunity to access resources and mates. You have better successful matings, and your genes will contribute more to the gene pool in future generations than those that don't fit those criteria, right? So you'll see that increase in allele frequency change over time. The Amish example. So this is where on the homework, like question 1, 2, and the last question, you'll kind of go back to this as an example, all right? So let's walk through it together, and then we can kind of deal with the cilantro, okay? So we've got an Amish sect, right? So a group of Amish that are living in one area. We have quite a few Amish living around us. We looked at 230 individuals, okay? 230 total. And 180 of them seem to have this sudden death syndrome, which results in them kind of having increased activity. Like imagine they're in gym class and then all of a sudden they collapse and end up dying. It's this terrible syndrome that they just recently found out about. For those that have just joined a little later, can you guys hop on the chat and fill in the link here? Code yourself for cilantro if you like it or if you don't. I don't like it, so you'll look at mine if you don't like it and code it the same way as mine, okay? All right, so again, if they have this kind of heart defect, it is a recessive trait, okay? So they're gonna be little a, okay? So how do we approach this? All right, so we approach it first, these recessive problems, by figuring out what the genotype frequency is for those that are homozygous recessive, or Q squared, little a, Q times Q, or Q squared, okay? To do that, you just take the number recessive and divide it by the number total, and that's your Q squared, okay? When you do that math, you get 0.783. You'll see that right here, right? 180 divided by 230, you get the 0.783, which is our Q squared, okay? That's the first step always in a recessive question. Figure out your Q squared. I'm given the Q, well, I'm given the number little a, I'm going to put it over total, and that's going to be my genotype frequency for the homozygous recessive. Okay, now if I've got a Q squared, I can easily solve for Q, right? What would I do, right? Square root it, right? If you square root that 0.783, you'll get your Q allele frequency. So that's the frequency of the recessive allele. Okay, now you have an equation that'll help you figure out what P is, right? P plus Q equals 1. Okay, so your next step after you figured out what your Q allele is to figure out what your P allele is. P equals 1 minus the Q, right? And when you subtract that, you get 0.115. Now, easy peasy. It's just a plug and chug scenario, okay? If I want to know, I'm going to go to the next slide. Everybody with me so far? Right? Good. Now, a lot of busyness on here, but super straightforward, right? If I ask you, how many are big A? How many are homozygous dominant? Just go to your genotype frequency equation. Homozygous dominant are the P squareds, right? I figured out what P was. I'm just going to plug in P squared times N, the total. And so my P was 0.115, right? I'm going to square it times my total. I get 3 individuals that were expected to be big A, okay? Now, if I want to know how many heterozygous carriers I would have, 2 PQ times N, okay? 2 times the P times the Q allele times the 230 total, I get 47. Okay, round to the nearest whole organism. All right, that's pretty important. All right, so now those are my two dominants, right? I've got my dominant homozygous, big A, big A. I've got my dominant heterozygous. I could add those two together and figure out how many were normal. 47 plus 3 is the normal, right? Now, how many recessives? Well, that was given to me originally in my question, right? That is helpful. That's a good way to check, right? Additionally, you can check yourself. That should be the Q squared, right? Numbers little a, that's Q times Q or Q squared. So Q squared times total, okay, gives me the 180. And so you can check yourself. The key at the end is add all three genotypes together and your total should equal the total given in the question. You guys good? All right, so well, let me just kind of finish off. Why is this important, right? Why might it be important for the genetic testing? And Kaylin had mentioned earlier that she had done the what is it, 21 and 27 and me, 21 and 11. What's it?
23 in May.
23 and me. Yeah, so, you know, getting that information can help inform you as you kind of reach your reproductive stage, right? You can make decisions. You could maybe go the route of, you know, working with a genetic counselor and in vitro, you know, like having them kind of seek an informed decision as you kind of move forward. In this case, in the case of the Amish, if they had known, this could be addressed by having a bit of a surgery, right, where you could implant with this, you know, ICD that would help kind of, you know, help their heart in times when this condition resulted. Because there's, I mean, the article that I read when this first came out was so sad. I mean, we had in one family 4 children that died of this disorder and they didn't know. They just didn't know. So it's kind of all coming to light. If you guys are interested, I think I hyperlinked the article. It's in here at the very beginning of that example. Okay. All right. So let's take a minute before I jump into the dominant. Let's take a minute. I'm going to put you guys into breakout rooms. Hold on. And here's the cilantro story. Okay. If you click back on the link, and I'll put it again in the chat, we're going to have our slide there. Okay. But let me set the stage here. We've got a recessive mutation, tastes like soap. So I am a soap taster. I'm a little a. Okay. And so we want to kind of work through the whole process and do what we just did on that assignment. Okay. Well, figuring out how many are big A, how many are little a, how many are big A, little a. Okay. It's a recessive example, just like we did with the Amish, right? First step should always be to figure out what your Q squared is. Okay. So given that, if I pull up our findings so far, okay, we've got 25 students that have been surveyed. That's our total. Remember the times N at the very end. We have 12 that are dominant. Now, 12 that are dominant, that they could be big A, or big A, little a, right? We don't know what they are. So we don't dive right in and say 12 over 25 is P squared. We don't do that. We always approach it through the only one way to be recessive angle. Okay. So we've got recessive here. Okay. We've got 13 that should be little a. Okay. Out of and 25 total. Okay. So 13 out of 25 is your Q squared, right? So I'm going to put you guys in breakout rooms in pairs. We'll come back in like, 5 to 6 minutes. Okay. And see if you guys have the same answers as me. All right. I just learned this myself. So we'll see how this goes. All right. I'm going to put you into breakout rooms. You can always go back to that slide. Okay. In fact, things you want to jot down, 25 total and 13 that are recessive, right? Little a. All right. Let me get you into those breakout rooms. Okay, here we go. Good luck. Join up. Rebecca and Sarah, are you guys there? Hey,
Hello.
how's it
All right.
going? Yeah, no problem. What's up?
Just have a quick question. Not exactly like what we're doing, but like what's the question you're asking? Like we're trying to figure out
You just want to
what
walk
about the
through,
cilantro.
just like we did with the Amish, right? So your first step needs to be, if you've been given number little a, or the number recessive, you put that over the total. So 13 out of 25 divided by 25 is your Q squared. Okay, so jot that down, right? Q squared equals 13 divided by 25. Now you square root it to find your Q, right? Kind of write the workflow down. Then you've got P equals 1 minus Q, right? Now you can find your dominant allele. Right? So far, so good?
I think so. So okay.
We'll
That should
go
be-
through it, I'm gonna pop it up here together. So
Yeah, I'm just trying to figure it out.
Q squared, it's, this is just you're on the fly, right? So, but if you had your PowerPoint open, that'd be easier. Q squared is little a, right? Q is the little a. So Q squared, little a, genotype frequency is going to be the number of recessives divided by your total. That's Q squared. You square root it to get your Q allele, the recessive allele, and then you can use your P plus Q equals 1 equation to solve for your P allele, the dominant allele. You just, do P equals 1 minus Q. Now that you've got your P and your Q alleles, you just plug and chug, right? If I ask for the number that are big A, that's P times P, or P squared. So P squared times what the total was. The total was 25, right? So you take your P allele frequency, you square it, and then you multiply that by 25. If I asked you what the number little a is, you take your Q allele frequency, square it, times 25. And then if I asked what your heterozygous carriers would be, that's 2PQ times total, right? So plug your P and your Q in, multiply it by 2, multiply it by 25, and you're left with all 3 of your expected number of individuals, right? Just like I have up here on the slide right after the cilantro. Shows you kind of the workflow. I'm gonna probably break everybody up right now so that we can kind of go through it, make sure everybody's good,
Okay.
and then we'll go from there. Okay.
Mhm.
Okay.
Is it just before you leave, is it that the so like the 13, the recessive, so that would be the Q? We'd put that in?
That's
If you know, don't like
that's
it?
the number little a. So you put that over 25 to get your Q squared.
Okay.
Okay. Okay, just like we
Okay.
just did in together
Oh,
with the other ones. Okay.
okay, thank you.
Yep, no problem. Are you dying? Are you dying to know? I was curious myself. Do we have everybody back? All right, well, again, I'll embed this. I'll update the PowerPoint so you guys can go back and look at it. I'll do it right after we get off today. If you go to the cilantro, I'm going to reshare again because I think you've lost the screen share. You can see the cilantro page. Okay. The next slide's got my answers, okay? And I'll repost that for everybody. And then here, the steps for the recessive question, okay? So when you're doing your homework, you can always go back to this, right? Our first thing to do is take the number on a recessive one, take your number recessive, divide it by the total, that's your Q squared. Square root it to get your Q allele frequency, subtract from 1 to get your P allele frequency. Then it's super easy. Then it's plug and chug. Number big A, number big A, number little a, you just multiply by your total and pull out the genotype frequency of each of those, okay? So you're just plugging in your stuff. Take a look at what I put here as I worked through it. So you guys, 13 of you were like me, don't eat soapy, right? So 13 out of 25 is your Q squared. Okay, when you do that math, you get 0.52, and when you take the square root of it, you get your Q equals 0.721. Most of you get that? Okay, subtract that from 1 to get your P. Okay, now we're easy peasy, right? Plug and chug for the expected numbers of each of these genotypes. So the number big A, that's a P and a P, or P squared, right? Take your P allele frequency, 0.279, square it, multiply it by 25. Only 2 people should be expected to be homozygous dominant, given what we learned from the class, okay? Now the big A, little a, heterozygous carriers, that's 2PQ, 2 times the P allele frequency times our Q allele frequency times the 25. So 10 of us should be heterozygous carriers, okay? Still loving cilantro, all right? And then we knew this already, right? In our Excel sheet, we had 13 that we knew were little a, but let's go through the math, right? 0.721 was the Q allele frequency, right? This is Q times Q, or Q squared. So 0.721 squared times the 25 gives us 13 homozygous recessive. Add all 3 together, the 2 plus the 10 plus the 13, check that you've got 25, right? And then thinking about it, 13 should be soapers. The other 2 are dominant. So add those 2 together. They're the ones that should love it. So, right, 2 plus 10 should be 12 that love it, and 13 soapers. So we're about even. Interesting. Good stuff. All right. I'm going to go back to John Travolta real quick. Super easy as well. You guys can see him? Okay. He's no Jason Statham. Anyways. So 75 out of 100 people, okay, are going to have these widow peaks, which is dominant. I don't have one, okay? If they're dominant, we don't know if they're big A, or big A, little a, right? That's a problem. So always, we go back to the default, which is the recessive. So the first step on a dominant is what I mentioned. You always want to say, you know, darn it, Danielle, or damn it, Danielle, she's given me one extra step. If I know that 75 out of 100 are dominant, I should be able to figure out how many are recessive by taking the total minus the number dominant, and it'll give me the number recessive, right? So if 100 is total and 75 are dominant, I'll do 100 minus 75 gives me 25 recessive as my first step. Then I just do exactly what you guys just did, do the whole process with the recessives, right? So then, now that you know the number recessive, pop it over the total, that's your Q squared, right? Square root it to get your Q on the next page, subtract from 1 to get your P. And then if you want to know how many are big A, little a, and little a, just plug and chug using the variables here from your genotype frequency equation. So the next slide will show you that, right? Numbers that are big A should be P squared times the 100 total. It's not always 100, right? That's just what we surveyed. A lot of people kind of default to the 100 and screw up the calculation, okay? So 25 are big A, heterozygous carriers. The P and the Q ended up being the same exact value in this case, and that's 50. And now we've got 25 for your homozygous recessive. We add all 3 together to double check that that's the total, okay? And you should be good to go. So again, this is just a slide kind of walking you through checking it, okay? But it's only one extra step. If I give you dominant question, You do total minus dom, gives you recessive, and then pop that recessive over the total, and that's your Q squared, and proceed just like you did with your recessive questions. Okay, so we did our cilantro mutant, and we'll go from there. Okay, so any questions on that? All that information we just went through should get you guys through your math homework. Okay, good practice. All right, so I want to briefly, kind of the last part of this lesson, just talk a little bit about fitness and genotypes. Some genotypes are more fit than others. So there's ways to evaluate that. If a genotype is more fit, we're going to look at the value W, and it's going to range from 0 to 1. And so one of the selection coefficient equations that you can use to determine if it's selected for is S equals 1 minus W, looking at your genotype fitness. Okay, nothing that I'm going to give you math on, just something to be aware of, that, you know, some genotypes are more fit than others. This is the important part down here in green. Okay, directional, stabilizing, and disruptive selection. Okay, imagine we think of a population, the length of fish, right? If we go and measure a whole bunch of fish, there's going to be some shorties, some long, and most middle range, right? Kind of a normal curve we would expect for the population. Now, what if there's some selective pressure out there, whether it's predators, hunters, fishermen, whether it's temperature change, right? So abiotic or biotic, competitors, right? Imagine there's something that favors one of those extremes, to be a big fish or to be a little fish. And because that selective pressure is there, over time, the ones that aren't selected for, like the shorties maybe, those will not survive in the next generation, right? The bigger ones would. They have an advantage. One of the extremes has an advantage. So over time, you'll see the gene flow, kind of the genetic population structure, such that you'll see more representative individuals with that genotype, right? And stabilizing means you select for the mean, so the medium sized, okay? And then disruptive means you select for both extremes, not the mean. It's better to have both extremes. That happens a lot when you have like unstable habitat, unstable environmental conditions, okay? So we'll look at some examples. This is what I was just talking about. You've got your normal curve, think of size, right? You line up according to height or weight. And if one of those gives you, is favorable in a given scenario, given environment, right, it will be selected for, and you'll see more of them in future generations, okay? If we have stabilizing, we see more of the mean. If we have disruptive, we see more of both extremes, less of the mean. And these are just some examples. Sheep horns on the sheep. If you're hunting the big ones, you're going to have less and less big ones in the gene pool. So you'll have smaller horns over time, predisposition for smaller horned population. Baby birth weight doesn't make sense to be big. Not helpful for the baby, not helpful for the mom, right? Or small, right? In terms of their health. And so that's usually a good example. And then disruptive selection is usually the beaks on the finches, okay? And so we give an example of directional with what happened with pollution. So back in the early days, industrial revolution, lots of, factories pumping out smoke and nasty sulfur, dioxide and whatnot, and it killed all the lichen on the trees. Lichen are, the mutualistic partner, the photosynthetic, the fungal partner, typically photosynthetic partner, and they died. They're very sensitive to air pollution. So as a result, they died, leaving behind not a light colored, because lichen are usually grayish, not a light colored bark, but a dark colored bark. So think about moths that are dark versus light. If it's very polluted and all the lichen go away and you're a dark moth, way to go, right? No predator is going to see you. You blend in, okay? And so here you can see when everything is polluted and the lichen are gone, being a black moth is a good thing. Being a white moth, a predator like a bird can find you, okay? Now come 1970s, when we start, really thinking about pollution, making changes, right, to reduce, the pollutants in the atmosphere, the lichen make a comeback and we see a shift. It's better to be a white moth as opposed to a dark moth. The lichen come back, the trees are lighter, and as a result, if you're lighter, you don't get eaten. And that's what we see here, right? The amount of sulfur, right? When there's more sulfur, we had more black moths. Here's the level of sulfur. As sulfur declines, we see less and less moths. And then it flip-flops when we have clean air, right? Very cool stuff. Disruptive selection, when it's dry one year and wet one year, we've got really hard big seeds one year, we've got small little seeds on another year. It makes sense for you to have different beaks that can chomp different size, okay? And as a result, you end up with having a couple of different phenotypes that are present because it's so unstable. Okay, we can see that with our beaks of the finches. Okay, you can see that some beaks are going to be smaller, some beaks are going to be larger over time. Okay, so beaks are usually the example with disruptive. And stabilizing, I mentioned, was sort of the baby birth weight. Here we've got tadpoles, okay? Tadpoles kind of following the same disruptive selection, right? We're looking at the frequency of the phenotype, we're looking at the progeny, and we're looking at, smaller tadpoles, small teeth, long intestines are herbivorous. Those that are larger, are going to be more carnivorous, and those that are medium size are just not good at eating much of anything. So we tend to have an example of disruptive selection as well. Now, just ending real quickly, guys, if you're really interested in genetics, there's some cool stuff going on with King Tut. They went and looked at the mummy, did some genetic analysis on it, and were able to determine that his parents were closely related, siblings, I believe. And as a result, he ended up with a clubbed foot, but he also had malaria, and that helped him not be able to survive a thigh injury. He broke his thigh, and so he ended up dying very young. So there's some cool stuff hyperlinked there if you're interested in his genetics. If you need extra help on genetics homework, if you're struggling, right, there's some examples that are hyperlinked here with an answer key as well, okay? And you guys can always reach out to me. But yeah, that's really the big take homes on the genetics, is to work through that math problem, the problem set, right? Do a couple examples with recessive, couple with the dominant, and feel comfortable with those concepts. Sound good? All right, we'll pick up with population growth on Wednesday. And if you guys can hop onto the quiz sometime in the next 24 hours, you should be all set.
Thank
All
you.
right. Bye, guys.